Fun. Now, there are other techniques that are more commonly used for separating big molecules. So I'm going to first describe a technique for separating nucleic acid molecules, usually DNA, although this will also work for RNA if we're careful. Okay? Large molecules, when we compare molecules in the cell, the largest molecules that occur inside of cells by far are the nucleic acids. Nucleic acids are way larger than proteins. Way larger than proteins. Okay? Consider that your chromosomes contain a total of 3 billion base pairs of DNA per haploid genome, meaning you have about 6 to 7 billion base pairs of DNA in every cell that you have. And nucleotides in general are considerably heavier than amino acids are. And a pretty good sized protein might have 1,000 amino acids. Okay, well, you can do the math. 3 billion is a heck of a lot larger than 1,000. And so the nucleic acids are way, way larger than the proteins are. So we have some different techniques for separating nucleic acids and proteins as well. Proteins are fairly large compared to other things that are present inside of cells. As I said, the first thing I want to talk about is DNA. Now, if you think about a DNA molecule, we haven't talked about structure yet, but everybody knows DNA is a double helix, and it's a pretty boring double helix. Okay? Beautiful but boring. It has, as part of it, the nucleotides that compose it. And then a nucleotide contains a sugar, a base, and a phosphate. No, you don't need to know that. Okay, we'll talk about that later. But the important thing is that every nucleotide in DNA contains one phosphate. Phosphate is a negatively charged molecule at physiological pH. So we can think of a DNA molecule as being a long polymer of negative charges, and that's exactly what it is. Every time the molecule grows by one more nucleotide in size, the charge gets one more negative charge added to it. So there's consistency with adding the nucleotides to the DNA and the change in charge. They go up in equal amounts. Now, this is very useful for the techniques I'm going to describe to you. So if I start and I have a mixture of DNA molecules that are of different sizes. Maybe I've got one that's 1,000 base pairs and another one that's 5,000 base pairs. I would like to separate them on the basis of size, but the difference in molecular weight between those, the exclusion limits on ion exchange, gel exclusion chromatography, don't allow me to do that. So I need something that's going to separate them better than what I can get with gel exclusion chromatography. And it turns out that this technology called gel electrophoresis is the perfect way to do it. Now, gel electrophoresis, I know many of you have probably poured gels before and run gels, looks like this technique right here. A gel is what I would describe as a matrix that has a composition not unlike Jell-O. Okay? It's a gel. That's what it's like. And that gel is made, typically, if this is an agarose gel, agarose is the material that we use to make this. Agarose is a polysaccharide that will dissolve in water when I boil it. And when I boil it, it's like making Jell-O. It's liquid, and as it cools down, it starts to absorb water and forms the matrix that looks sort of like this. There's sort of a mesh of these polymers of agarose that are in there. This mesh has, if I mix it up nicely, reasonably consistent size of holes that we can think of through which the molecules will pass. There's little holes there that these guys are going to pass through. And in our case, the molecules are DNA. They're not showing DNA on here, but ours are DNA. Well, DNA is, as I said, double helical. It forms a nice rod. It's flat. It straightens out like a rod. It's a polymer of negative charges. And so if I take my mixture of DNA and I apply it to the top of this gel, as you see here, they will sit there, the mixture will sit there in one of those little wells until I apply an electrical current to it. And when I apply an electrical current to it, what happens is I have a plus end and I have a minus end. And so if I hook my gel up properly, and believe me, everybody who's ever run a gel has hooked electrodes up the wrong way. But if you hook them up the right way, You have the positive electrode down at the bottom, the negative electrode up at the top, and your DNA is up at the top. Well, negative repels negative, and positive attracts negative. So the DNA molecules get literally pulled through, or pushed, as you want to think of it, either way, through this matrix of material. The small guys have the easiest mobility of moving around, and so they're able to travel the fastest. The big guys are much more bulky. They have a much more difficult path to make it through this gel, and they travel more slowly. When I run a gel like this, you can start to see the separation that's here. Here's a mixture of 3 different sizes of molecules, green, red, and yellow. And you can see the little bitty reds are the fastest, the medium-sized greens are the next fastest, and the big yellows are stuck up in here. Okay? This is very useful, and let's see. That's not the gel I wanted to show you. I thought I had another one. No, I don't. Okay. Well, suffice it to say that if I run a gel like this, I can separate the individual bands of DNA on the basis of their size, and the separation is pretty darn good. I get bands that come across on the gel, and if I run what's called a ladder is a set of standards of known size, where I have something that might be 10,000, 8,000, 5,000, 2,000, 500, something like that. I can look where my material ran compared to where the standards on the ladder ran, and I can very easily say, okay, there's my 5,000 band, there's my 1,000 band, etc. And so I can actually size my nucleic acid very easily if I do that. So, now I tell you, I start with DNA for electrophoresis because DNA is the easy one to understand. DNA has 2 properties that make it very easy to do electrophoresis. One, as I said, is it forms a sort of a rod. That is, it has a consistent size. It's not globular like a protein. And second, it has that consistent negative charge. Now, the proteins that I've told you about don't have those properties. Proteins are all mostly globular. There are some fibers, but mostly globular proteins. And some of them may have a positive charge. Some of them may have a lot of positive charges. Some of them may have negative charges, some of them may have neutral charges, etc. So we've got a real mix of charges. If I want to use gel electrophoresis to separate my proteins in a mixture, I've got to make those proteins look something like the DNA. Okay? So that's the number one thing, is I've got to make those proteins look something like the DNA. Second, proteins are smaller than DNA is. So the agarose gel that I poured for DNA isn't going to work very well for proteins because they just come shooting through. The holes are so big that all the protein molecules will make it through very easily. So instead of using agarose as the material for my gel, which is what I use for DNA, in the case of protein, I use something called polyacrylamide. Now, your book goes into the chemistry of polyacrylamide, and that's not really important for our purposes. What matters with polyacrylamide is polyacrylamide starts out as monomeric units of acrylamide, and they can be polymerized together by adding a chemical. And so when you polymerize these guys together, they form a mesh, not unlike what we saw with the agarose, but now the mesh is such that the holes that we saw before are much smaller. And that's important for our separation. So polyacrylamide gel electrophoresis, it's called PAGE, polyacrylamide gel electrophoresis, will allow me to separate proteins if I make those proteins like nucleic acids. I'm going to tell you how to do that in a second. So polyacrylamide gel electrophoresis is going to take and make that matrix of support with those little holes, and the holes are going to be considerably smaller than they were with agarose. Okay, well, how do I take and make a protein like a nucleic acid? And that's a real problem, and the trick that we use to separate proteins this way is very, very clever and very, very cool. Okay? It involves using a detergent, first of all. Now, I haven't really talked much in class about detergent. I mentioned in the highlights that detergents have the property of denaturing proteins, and they denature proteins by interacting with their hydrophobic regions. In fact, detergents will interact with an awful large portion of the backbone of a protein, including the polar as well as the nonpolar regions. If I think about a globular protein and I denature it, denaturing should tell you, first of all, that it's changing the shape of it. Okay? It's changing the shape. Second, detergent is composed of fatty acids. So I have a nonpolar end and I have a polar end. Okay? A nonpolar end and a polar end. And it turns out that if I mix a detergent called sodium dodecyl sulfate, which we more commonly call SDS, if I take and I mix SDS with a mixture of proteins, each protein will literally absorb those SDS molecules. The protein will denature, and when it denatures, it becomes a flat rod. It lays out as a flat chain. Now, that brings to mind something that happened with the nucleic acids, because the nucleic acids already started out as a rod. In this case, I've taken the protein, and I took this all knotted up mess, added some detergent, and it relaxed and laid out as a nice flat rod. That rod is coated with the detergent. It's coated with the detergent. Now, detergents, in the case of sodium dodecyl sulfate, most detergents, at least all detergents made from fatty acids, have a very useful property. The useful property is that the polar ends that are all sticking out are all negatively charged. So now, by just simply adding SDS, I have made the protein structurally look like DNA at first glance. It's a rod, and it's a rod that has negative charges sticking out. The longer the rod, the more negative charges. Just like the longer the DNA molecule, the more negative charges. So adding SDS to the proteins have converted the proteins now into a form that I can separate by polyacrylamide gel electrophoresis. Everybody understand that? Yeah. What does the SDS stand for? Sodium dodecyl, D-O-D-E-C-Y-L, sulfate. Yes, Phil? It will interact with the hydrophobic regions, but as I said, SDS will interact with basically all regions of the protein and basically just cause it to lay out. That's what it's doing.
So it's pretty much parallel with DNA in the fact that it just has negative charges on the outside.
Exactly. Exactly. Okay? All right. So now I've got all my mixture of DNAs. I add it to the top of an SDS, what's called SDS page column, okay? Because SDS is going to be critical for this separation that I want. So I do SDS page. I can hook it up exactly as I did before, because now all of the proteins are negatively charged. All the proteins are rods. And I'm going to be separating solely on the basis of the size of the individual proteins that are there. So I hook it up so that my negative electrodes at the top, my positive electrodes at the bottom, and the proteins go racing through, the smallest ones going first, the biggest ones going last. And when I do that, I get something like the gel I showed you earlier, which was right here. Now, this is a protein gel. It's SDS page. And what you see is on the left and right sides, you see what's called that ladder that I talked about for DNA. A ladder is simply a set of molecules of known size that I can use to determine the molecular weight of something that I don't know. Now, in this case, there's only one protein. This was a purification that somebody was doing, but there was only one protein that was on there. You see it right here. If I took all of the proteins of a cell and I ran them on a lane, that is, I hadn't purified them, what I would see are hundreds or thousands of bands in a given lane. Okay? So that's what will typically happen when I first bust open cells. Now, this has obviously been purified quite a bit to get to this point. Okay. Question? Okay. Yeah. In real life, it's used considerably, actually, when you're trying to purify a protein. One of the things that you'll do is to monitor the progress of the purification. So as I noted, that you wouldn't normally have one step and then you had a purified protein. You would have to go through several things. And so at each stage, you would want to be monitoring how much of the particular band that you have compared to all the other bands that are present. So in the real world, you use it a lot, an awful lot. Yes, sir? That's correct. So SDS page is normally used with a mixture of proteins, sorting out and seeing what's there. Okay. Okay. All right. So that gets us, there's sodium dodecyl sulfate, if you want to see what that looks like. Okay. You don't need to know the structure of it, but it's, as I said, it is a fatty acid with a long chain that has, in this case, a sulfate placed on the end of it. Now I want to turn to a technology that's going to use polyacrylamide gel electrophoresis, that is SDS page. Okay. By the way, if I say PAGE and not SDS PAGE, that's a different thing, right? If I said you ran a bunch of proteins on polyacrylamide gel electrophoresis but didn't have SDS, what do you suppose would happen when you turn on the electrodes? They would go both ways, right? Because some of the ones that are negatively charged are going to go in, but the ones that are positively charged are going to go boing. They're going to come up, okay? And sometimes we actually do run what are called native gels, and there's reasons for doing that, and I won't go into those here. But there are sometimes we do native gels. So polyacrylamide gel electrophoresis without SDS is another technology. Okay. Well, so the technology I'm going to describe to you uses SDS page as part of its procedure, but it's the second part of the procedure. So what I'm going to describe to you now is another gel technology that involves two steps. And you have to understand the first step before you understand the second step. So it's kind of like dancing, I suppose. Anybody ever take the two-step dancing? No. Okay. Has anybody ever done anything? No. All right. Two-dimensional gel electrophoresis has, as I said, two components. The first component I'm getting ready to describe to you now. The second component involves SDS page. Let's imagine I have my cells I've taken and I've busted open, and I have that mixture of proteins. You know that proteins differ in amino acid composition, and you know if you work any of the problems that I have assigned for you or you've looked at, you'll see they have different charges as well. Not surprisingly, they have different isoelectric points. Okay, that is the PI. The technology of two-dimensional gel electrophoresis uses as its first step something called isoelectric focusing. Now, isoelectric focusing is a remarkable technique for separating molecules on the basis of their PI, that is their isoelectric point. So the separation is on the basis of their PI. How does that work, you might wonder? Well, what you do is you take a tube of material and you put into this tube something called a mixture of amphilites. This mixture of amphilites has some molecules in it that have long polymers, long chains of negative charge. It has some molecules that have not quite so long, not quite so long, not quite so long. And then it has some that have short chains of positive, longer chains of positive, longer chains of positive, etc. So we've got a mixture of things that have a lot of negative charge, a lot of positive charge, and a lot of things in between. You with me? If I take that mixture in that tube and I apply an electrical field to it, the things that are the most negatively charged are going to go over here towards the positive electrode, and the things that are most positively charged are going to go over here towards the negative electrode. And the ones that's going to go the fastest to the negative electrode will be the ones that are the most positively charged and have the most positive charges in there, and vice versa on the other side. In between, they'll have fewer and fewer and fewer and fewer and fewer and fewer. Until I get in the middle where things I have zero. Everybody with me? Now, I'm not going to go through the chemistry and show you, but what I will tell you is if I apply this electrical field and I let it sit and get to equilibrium, what happens is that I create a pH gradient in this tube. From a very low pH to a very high pH. A pH gradient in the tube. You could imagine these things pick up and give off protons in very different manners. Okay? And so that's what I've created by making this electrical field. When I create the field, instead of just taking just the amphiolites, I take my mixture of proteins and put them in there at the same time. Here's what happens. The very positives over here, that is the amphiolites. The very negatives over here. So I have a pH going from very low to very high. And that means if I go from 0 to 14, I've got every pH in between represented. When I apply my electrical field, I've got a mixture of proteins. My protein that might have a pI of 4 starts moving over here until it gets in the region where there's pH 4, and it stops moving. Why does it stop moving? Say what?
Reach equilibrium.
Why does it reach equilibrium? What's equilibrium mean?
Well, the exchange of
Well, you're not quite saying what I want you to say. You're on the right track.
The negative and positive are pulling equally.
Okay, he says the negative and positive are pulling equally, and well, that's true too, but there's a much simpler reason why it stops moving. What's the PI mean, folks? It's isoelectric point. What's isoelectric point mean? What's the definition of PI? Uh-oh, pop quiz time. Somebody tell me. What's the definition of PI? No, it's not the charge of 0. What is it? The full definition. What's that? No, not the overall charge. There's a definition, folks. Is something always charged 0? The pH at which the charge is 0. If you tell me on an exam that the PI is when the charge is 0, you're going to be wrong. The PI is the pH when the charge is 0. This guy moves over here to pH 4. It's at its isoelectric point. It no longer has a charge. It's not going to move in an electric field. Okay? So the molecules in here, if they have a PI of 4, are going to be separated from molecules that have a PI of 5, are going to be separated from molecules that have a PI of 7. In fact, with this technique, I can separate molecules that differ in PI by 0.01 pH units. It's very precise. Okay? So I've just done the first part of my technique of 2D gel electrophoresis. I have separated in this tube my mixture of proteins on the basis of their PI. Do you suppose all these proteins are the same size? No, definitely not the same size, because I already told you if I took a mixture of proteins and I run them on a lane, I'm going to get thousands of different sizes. But I've separated them on the basis of their PI. Okay? If I now take that tube of material and I cut a little hole in it such that the tube of material sort of oozes out onto the top of an SDS page gel, what I have is a situation that looks like this. The bottom portion of this is my SDS page gel. Here's my tube of material. The proteins have been separated on the basis of their PI. Now I run SDS page running down this way. And yes, I had to add SDS to that after the fact. Okay? After I did the PI separation, I added SDS. Now I'm going to do SDS page, and I'm going to separate these proteins on the basis of their size. Now this is very cool. When I look at my final product, it's going to look something like what I see schematically here in this image. I'll show you a real image in just a second. Going in the up and down direction, I have the biggest guys at the top. Going in the left and right direction, I have the lowest PIs on the left, the highest PIs on the right. So if I were to compare, if I were to ask you on an exam, this red guy right here compared to this orange guy right over here, what would you be able to tell me about it? I would hope that you would be able to tell me that it is, in fact, larger and it has a lower PI than this guy does. Now this technique is phenomenally powerful. Because for the first time, researchers with this technique are able to separate every single protein in a given cell. Every single protein. What does it look like in practice? It looks like this. Now, it looks like a real mess to you, but if I take a known protein and I run it on SDS page and I compare it to this and I say, oh, look at that guy right there. That's a DNA polymerase. And I know where DNA polymerase now is in this mixture of stuff. I know how much of it is by how dense the spot is. It's very, very useful. Let's imagine I'm a cancer researcher. Let's imagine that I've got a patient who has a tumor in their colon, for example. Okay? I isolate some of this tissue. I grind up this tissue. I get rid of all the other junk by centrifugation, and I say, okay, let's do SDS page. I take and I run, and I get a pattern that looks like this. And I say, that's what this looks like in this cancer patient. That's not very useful by itself. What if I take tissue from a normal patient from the colon, from the same region, and I grind it up and I do that isolation, and now I've got my cancer 2D plot versus a normal 2D plot. It becomes very easy for me or for a computer to say, what's the differences? Ooh, look, this spot's found in the cancer patient, but not in the normal patient. I've all of a sudden identified a protein involved in a tumor. Okay? So this technique is extraordinarily powerful, and it's used to analyze what's called complete cell extracts. And these complete cell extracts tell us a tremendous amount about what cells are doing. A tremendous amount about what cells are doing. Okay? So this is very useful. I could use this in other ways. Imagine that I say, I wonder what the effect of somebody taking codeine is. Okay? Codeine causes some people to get sick. Okay? It's a painkiller. It causes some people to get sick. Same experiment. Person not taking codeine, person taking codeine. Compare tissues. What's the difference? Okay? This is very, very powerful. Okay. I'm sorry? What would it cost to run one of these? It's not prohibitively expensive. You would spend more on paying the technician to do it than you would spend on the materials, which is not uncommon in science. The gel material is fairly trivial. One of the issues that you have with this technology is actually getting it as reproducible as possible. How far do you run it here? How far do you run it here? And so forth. But that's gotten immensely better. And so if you get it in some good hands with good people, they can do the same, virtually identical thing a couple times in a row, which is good. Yes? It actually looks like this. The gels themselves will be about this big. So, of course, this is blown up on the screen. But, you know, by maybe, what, a couple feet by about 1 foot high. Very powerful technique. Okay. Okay, time is slipping away from me here. All right. Now, what I hope I've impressed on you as I've gone through this is the fact that purification does not occur in one step. And if you've ever purified proteins, you know that, and you know that you have to be a little creative in your use of techniques sometimes to get a protein pure. One of the things that's essential for you to do during that purification process is to monitor how much activity you've got. Why do you want to do that? Well, you want to do that for a couple of reasons. One, remember how I described that sometimes you don't want to throw away something that you, oh, I know it's not in there, and I throw it away and discover that's where it was. So one of the things I'm going to do is whenever I do a step, I'm going to monitor how much of the material I still have in that step. Okay? And so when you do that through a variety of steps, you can produce data that looks a little something like this table right here. And let me just step you through it. If I take a tissue like this colon tumor and I grind it up by homogenization, what I discover is that I measure, first of all, the total amount of protein that I have in there. And that's fairly easy to do. And I do that to just kind of monitor my progress of purification, as you will see. I know what my protein does because I can add a chemical to it and watch how much that chemical changes color, so I know how much activity of my protein I start with. That's useful for me as well. So in this case, I had 15,000 milligrams of protein, and I had 150,000 units of activity. And a unit is simply a measure of how much protein I have. It might be absorbance, it might be radioactivity, a variety of things that it might be. But for our purpose, we're just going to call it units. The specific activity of my protein will be the total number of my extract will be the total amount of units divided by the total amount of protein that I had. Now remember, the total amount of protein that I have to start with is not only my protein, but all the other proteins that are there. So that 15,000 milligrams is not going to be all my protein. That's going to be all of the proteins from the cell. So I start with 10 units per milligram of specific activity, and that's the definition of specific activity, units per quantity, in this case the quantity being milligrams. My yield is 100% because I haven't lost anything. I haven't really done anything except homogenize. And my purification level is 1, and I'll explain that to you in just a second. After I take, and I haven't explained salt fractionation, but that's just another technique. After I take and I salt fractionate my mixture, I discover I only have 4,600 milligrams of protein left. But I still have 138,000 units left. I've gotten rid of 2/3 of the protein, but I still have about 90% of the activity. That's not surprising because I'm purifying. I'm wanting to throw away the bad and keep the good. And that's what I'm doing, basically. My specific activity in this case is this number, 138,000, divided by my total milligrams of protein, or 30 units per milligram. My protein has gotten purer. More units per milligram means my protein is purer. My yield at this point is 92%. And what does that mean? My yield is how much activity I have now compared to how much activity I had to start with. This divided by this times 100. My purification level is the increase in specific activity that I see. That is the 30 divided by the 10. I go through various steps. Ion exchange chromatography, it gets purer, and we can see the specific activity going up. Gel filtration chromatography, it gets purer, and we see the specific activity going up. And finally, after affinity chromatography, look what's happened. I'm down to 1.75 milligrams of total protein. I've thrown away almost 15,000 grams of junk, milligrams of junk. I still have 1/3 of my activity. That's great. That may sound like, well, you lost a lot. But in fact, that's very, very good because I've gotten rid of an awful lot of stuff I didn't want, and I didn't lose many babies with the bathwater, if you want to use that analogy. I kept most of the babies. Okay? My specific activity went skyrocketing. This activity divided by this amount of protein, 30,000 units. My yield is 35%, which is this number divided by my starting number up here. My purification was 3,000-fold. It's 3,000 times purer than it was when I started. Okay? Now, these sorts of things we have to monitor as we're purifying a protein. And yes, I do think this is a calculation that you should be able to perform. Okay? So you should understand how these numbers relate to each other and how you calculate them. You never know, you might have to do it on an exam. Okay. That was the gel I was looking for earlier. Here's a purification. In fact, this is described as protein. It's just a schematic of protein. But in fact, this looks very much like a DNA gel, what a DNA gel will actually look like. And well, actually, I should point out, what they're doing here is they're showing you what it looked like at each step. Here's what you started with. Here's after the first purification. Here's after the second. Here's after the third. And here's after the fourth. So here's a real good example you asked for in the real world where you did that. This is a real world example where you would monitor how much of this material. And you can see this starts out with a very tiny amount, but you're getting more and more as you get towards the end. Yes, sir? Good question, Casey. Can you extract this from the gel? You can, but that would be a good exam question. Okay. I'll tell you what. There's an extra credit exam question. There's something for you to. I like to do these. Something for you to think about. Okay. and you can think about it. I don't want you talking to each other. I don't want you asking advice of others for this, so don't go to the TA and say, what's the answer? Honor system here. But the idea is, when would you feel comfortable taking this from a gel and working with it? And when would you not feel comfortable taking it from a gel and working on it? Okay? Extra credit question on the exam. Okay. Now, I'm not going to talk about S values. We'll talk about those later in the class. And what I want to do now in the remaining time that I have about 5 minutes, is I want to talk about some ways that we can treat proteins. Okay? We have to treat proteins in various ways. We chemically modify them so that we can purify them, so that we can analyze them in other ways, etc. And one of the most common modifications that we do to proteins is to break them into pieces. Break them into pieces. Proteins are smaller than DNA by a long ways, but proteins are still pretty good size. 1,000 amino acids is a pretty complicated molecule. I can't take 1,000 amino acid protein and work with it very well. My analytical techniques for determining size, which I'll talk about in the lecture on Wednesday, don't work on molecules that big. So if I want to work on it and I want to understand it, I really need to break it into smaller pieces. And so I have to use agents that are that do what are called proteolytic cleavage. Okay? The first one that I want to describe to you is a chemical. This chemical is called cyanogen bromide, which is also abbreviated CNBr. And cyanogen bromide has the property that wherever there's a methionine inside of the protein, cyanogen bromide will break it on the side where the carboxyl group is. Okay? It will break it on the side where the carboxyl group is. So we can see here, here's the methionine. When it's treated with cyanogen bromide, we make this ring structure, and we break that bond that was right here. Okay? I'm sorry, the bond was right here. It's right here. Okay? So we break this bond right here. That releases that. So anytime I have a methionine inside of a protein that I'm studying, I can cause a break to occur by treating that protein with cyanogen bromide. That is a proteolytic cleavage. It's a chemical cleavage, and it breaks a protein into smaller units. Another cleavage agent for breaking proteins, and these are very common, is something known as a protease. A protease is an enzyme. Whenever you hear the letters ASE, as in A's, at the end of a name, it's always an enzyme in biochemistry. Protease is an enzyme that breaks a protein down. There are a variety of proteases, and what these proteases do is they catalyze the breakage of peptide bonds. And they generally cause breakage to occur at specific places in proteins, just like cyanogen bromide worked only at methionine residues. Proteases have specificities, that is, specific amino acids that they will break at. The protease known as trypsin will break the peptide bond on the carboxyl side of lysine and arginine. Okay? So we take and we break right here. If this is a lysine or arginine, this bond will be broken. So again, we have agents for breaking proteins at specific places. Okay, that's a good stopping point. I finished talking last time rather hurriedly about agents that we can use to break polypeptides into smaller pieces. And I want to emphasize that when we choose to do this, we don't want to randomly break something into pieces. We can imagine that we could do some sort of a chemical treatment that would randomly break things into pieces, but that actually turns out not to be very useful to us. So we want to break polypeptides into specific pieces, and so having reagents that will cut at specific places is very important. So that very first one that I mentioned to you, which was cyanogen bromide, is a chemical agent. But it's a chemical agent that works in a specific place, and as I mentioned, it's on the carboxyl side of methionine residues. So we know when we treat a protein with cyanogen bromide that we're going to cut it at every methionine residue. Similarly, I briefly mentioned to you that we can take and cut with an enzyme known as trypsin. And when we cut with trypsin, we cut it at lysine and arginine residues. And again, on the carboxyl side, as I noticed, that means it's going to cut right there between those two bonds. There is a table of polypeptide cleavage agents shown here, and no, I'm not going to ask you to memorize the table, but there are some cleavage agents that I think that you should know where they cut. Certainly cyanogen bromide is one of them. Trypsin is another one of them. Chymotrypsin is another one. Okay? And so you should know where these different things cut. And the last one is carboxypeptidase A. And I'm going to make carboxypeptidase A easy for you. We're going to assume that it doesn't have specificity. It only cuts at the very terminus. All right? And so what it does is it cuts, if I were to show you a polypeptide chain looking something like this, the way that carboxypeptidase A works is it's the only one that does not have specificity. It starts at the carboxyl end and clips off an amino acid. And then it clips off another one. And then it clips off another one. So sequentially, one by one by one, carboxypeptidase A will clip off amino acids from the carboxyl end. Okay? Now, there is some specificity to the enzyme, but I think it just complicates the case for us. So we're just going to treat it as if it cuts equally at all amino acid residues, starting at the carboxyl end and moving inwards. Okay? So that's something those are reagents that I think are useful to know. And I don't ask you to memorize it just for the sake of memorizing it, but these are polypeptide cleavage reagents that are very, very commonly used. So it's useful information for you to know that. Okay, now I'm going to skip over several things on the outline, and one of the questions I always get is, do we have to know if you don't go over it? Okay, and I will tell you that with the exception of the video that I did on the PIs and polypeptide charges, unless I talk about it in class, you're not responsible for it. Okay? That video that I posted you are responsible for, and I didn't talk about that in class. But if I skip over something here, I'm not going to come back and dig it up and make you guys learn it. Okay? I think if it's important enough, I should talk about it. And I also think that if I talk about it, it's important enough that you should know it. So it sort of goes both ways. Okay. Now, what do I want to say? Disulfide bond reduction. Okay. One of the reagents I told you about that we can use to reduce disulfide bonds in polypeptides is mercaptoethanol. Another reagent is dithiothreatol, which is shown here. You don't need to know the structures, but you should know the name. You may know it better by the abbreviation. It's frequently called DTT. That's DTT, not DDT. Okay. And it works very similarly to mercaptoethanol. One of the things that we want to do sometimes when we're studying a protein is to block the ability of the sulfhydryls to come back and form a disulfide. And there's various reasons we may want to do this. We may be analyzing the amino acid sequence or a variety of things inside of the protein. So once we reduce it with a mercaptoethanol or a dithiothreatol, we want to block those sulfhydryls and keep them from coming back together. There's a couple ways of doing that. One of the ways of doing that is by treating the sulfhydryls with iodoacetate. And when we treat with iodoacetate donates this acetate group onto there, and when the acetate group is sitting on the sulfur, that sulfur is no longer reactive and can no longer form a disulfide bond. Okay, so that's one way of preventing those the sulfhydryls from reforming disulfide bonds. Okay. Another reagent that we can use to break disulfides and keep them from reforming is to treat the disulfide with performic acid. Performic acid actually oxidizes the disulfide bond and creates cysteic acid residues that you see here. And these cysteic acid residues will, in addition, not come back and form a disulfide bond. So there's a couple of choices that we have then to prevent disulfide bonds from forming. Okay.